and you may wonder what are residents? Why do you have them? Part of the motivation to have residents in BrainHack School is that you're doing the project, working together, that's all great, but we are also trying to have a number of instructors to spend time with us, and I wanted those people to be able to act too, and do projects too if they want to spend, a couple of days or even weeks with us, just doing their thing. And the idea with the resident was really like to bring people, I wanted to spend time with, basically. So anyway, Claire, because it was way too long that we didn't have time to chat for a long period of time. And the other motivation for residents is more like inspirational figures, people who I think can be role models for what is it we're trying to achieve with the BrainHack School. And that's true for all the instructors that we invite to come, especially the students who I think are really amazing and pushing the envelope of best practices in the field. But definitely our residents are there for it too. And so when I met Claire, I was doing some kind of like functional connectivity analysis and parcellation, which means like cutting the brain into little pieces that would be homogeneous. And I was trying to understand if that made any sense. And it didn't. Like I was kind of like, I had those parcels and I didn't know what to say about them. And it was really like a sort of prototypical case of a guy coming more from computer science, touching your imaging data and having no clue. And so you run the algorithm, you can test if it's reliable, you can test if it looks good, but after that I felt like I was stuck. And in the meantime, Claire was working with Mike Petridis, who's an expert in neuroanatomy, and really looking at a few regions. Well, I was looking at the full brain, she was looking at the insula, she was looking at bone marrow areas 44 and 45, really doing very careful work to look at all the voxels cells and trying to relate them to the known anatomy in humans and in monkeys. And in a way, she was mixing the kind of multivariate methods I was using with actual knowledge in neuroscience. And her stuff made sense. Her stuff was convincing, and her stuff really brought understanding to the things that I spent years trying to understand. And so in terms of the BrainHack School, trying to mix field and really living at the intersection of neuroscience, statistics, computer science, hacking, I think she's, for me, a role model. She really inspired me in my work and seeing how you could actually navigate that field and try to make sense of those complex data. So I felt she was probably one of the best people to invite. So thanks so much for accepting. And now I'm going to let you do your thing.
Thank you, Pierre. I'm incredibly flattered, and I feel very lucky to be here and be part of this school, which I just think is the most incredible opportunity. I think things have changed so much since I was a PhD student and even early postgrad or postdoc. And the kinds of skills and the kinds of skills you're gaining, the kinds of exposure that you're getting in this school are going to stand to you for your entire careers. And I'm really bowled over at people's knowledge and their abilities already, even so many of you are at such early points in your careers. So thank you very much, Pierre, and thank you everybody for giving me this opportunity to talk about my work. So this is going to be quite an overview, a scientific talk rather than a methods talk. And Pierre really pushed me to include more recent work. So there'll be a lot of preliminary data and some hand waving and some things that need to be taken with a pinch of salt because they're at quite early phases at the moment, but just to give you a sense of some of the directions that I'm trying to go in. So my work uses fMRI, so functional MRI, to examine, to learn about brain functional organization, how it changes over development, and how disturbances in typical brain development can give rise to psychiatric disorders. So the ultimate goal of better understanding when, why, and in whom psychiatric disorders emerge. So with that goal in mind, I'm going to start by talking about some of the challenges that we face when we try to use fMRI to particularly the conventional task-based fMRI approach to study young children, brain function in young children, and in those with neurodevelopmental disorders. I'll go on then to describe the functional connectivity, or to use the fancier term, functional connectomics approach, which I think it's fairly fair to say has really revolutionized functional neuroimaging over the past 10 to 15 years. So I'll talk about some of the advances that this approach has allowed us to make in terms of our understanding of brain functional organization and of its development and how it's disrupted in psychiatric disorders. And then I'll go on to talk about the direction that my work is taking that aims to leverage this approach to, I suppose, build better biomarkers or improve the sensitivity of biomarkers for neuropsychiatric disorders, particularly those that emerge during development, which I think is most psychiatric disorders. So I'll talk about some of our efforts to improve the sensitivity of behavioral measures as well as neuroimaging-based measures of child and adolescent brain function. And at the end, I'll turn to the direction that I'm really quite excited about, which is translational approaches, which I think are, where we have a real opportunity to develop a mechanistic understanding of how brain development is disrupted in psychiatric disorders. Okay, so I'm sure at this point everybody's fairly familiar with MRI and probably even the conventional task-based MRI approach, where essentially what we are doing is measuring changes in brain activity over time and trying to map those changes to changes in cognitive state that we have elicited with a particular kind of task, right? So in this particular example, it's a lexical decision-making task, so participants just have to indicate which of the stimuli is a real word as opposed to a non-word. And when we analyze these kinds of data, we, in its simplest form, we might have blocks of 30 seconds where a participant does the task, then 30 seconds where they rest, 30 seconds task, 30 seconds on, 30 seconds off, and so on. And when we contrast the periods where they're doing the task with the periods where they're not doing the task, we get these nice statistical maps of, evoked brain activity, and in this case, mapping out the left hemisphere-based language network. So, of course, this task-based fMRI approach was the dominant approach in the field from its inception about 25 years ago or so, and has given us enormous insights into brain function and brain functional organization. But you can appreciate that this kind of approach will work pretty well with adults, and pretty well probably with older adolescents who are capable of lying in the scanner for long periods of time, staying perfectly still and doing this kind of pretty boring task. But what if we want to study periods of development where arguably the greatest kinds of changes are taking place, where cognition is really changing quite rapidly? Then we're going to want to look at younger kids, maybe 6, 7-year-olds. If we're interested in language development, we probably want to look much younger, maybe 2-year-olds, or maybe even from infancy, right? And of course, we can't do, none of these populations can really do this kind of, can be studied with this kind of conventional task-based approach. Although people have, tried to do younger kids, it's really, really challenging. What if we want to do translational work? What if we want to work with monkeys or rodents? Then we need a really different approach. We're not going to be able to use traditional task-based methods. And so this is why in the last 10 years or so, we've seen this huge explosion of interest in what has been variously called a resting state functional connectivity or just functional connectivity approach to studying brain functional organization. So it involves collecting just exactly the same kinds of data that we collect during tasks, except this time, most of the time, we just have participants lie in the scanner with their eyes open, not doing anything in particular. And this is part of what's so powerful about that approach, because as you can appreciate, this makes we are able to collect this kind of data from a much wider range of populations now, because these are the lowest possible demands. They just have to get in the scanner for even as little as maybe 6 minutes while they relax, rest, stay still. They can be sleeping, they can be sedated, people can be under anesthesia. We can collect these data from anesthetized or sedated animals. We can look across a broad variety of conditions and now a much wider range of populations, including those who have very low cognitive ability or capacity. They don't even have to be looking at a blank screen or sleeping. Increasingly, people are using movies like Despicable Me because, particularly for young kids, these are engaging, they keep them still in the magnet, and we can still get the same kinds of data. We can still look at functional connectivity in the magnet using these data. So what does this resting state data give us? How do we do these analyses? What does it look like? So one of the nicest illustrations is the very first study, which was performed by Bharat Biswal back in 1995, where he had 11 participants go into the magnet and perform a really simple finger tapping task. So he just had them tap their fingers at their own pace for 30 seconds, then rest, tap again, then rest, and tap. And when he mapped out the task-related activation associated with this task paradigm, he saw this really nice map of activation in primary and supplementary motor cortex. He had the same participants in the scanner not do anything at all for 5 minutes. And he took, using this now resting data, separate data, and I believe he counterbalanced the conditions so they weren't always doing the finger tapping first. But he took a voxel located in primary motor cortex, so the area that had been activated by the task, and he simply extracted the time series, a pre-processed time series, from the resting state data, performed a really simple correlation, asked what other voxels in the brain show a pattern of activity in this resting state, so just intrinsic or spontaneous activity that's correlated with this voxel in motor cortex. And what he saw was a map that bore remarkable resemblance to the pattern of task-evoked activation. So this coordination or synchronization of spontaneous activity is what we call functional connectivity. Bharat's study was met with enormous skepticism and essentially ignored for 5 to 10 years until other people started to pick it up and started to demonstrate that this phenomenon wasn't specific to sensory motor cortex, but could be detected across the brain in any functional network you can think of. So language, memory, visual, any functional domain we can think of could be mapped out in the same way. So here's another example of that. Again, it's one of my earlier studies. This is the one we did with Mike Petrides, where we were mapping out the functional connectivity of Brodmann's area 44. And when we put a seed in area 44, map out the regions in 24 participants who are simply resting in the scanner for 6 minutes, regions that are correlated with that seed region, what we get is this lovely map of left hemisphere based language network. And it maps really well onto, in this case, the map produced by a meta-analysis of over 128 lexical processing tasks studies, so involving hundreds of participants doing the kind of task like I mentioned at the beginning, like deciding whether things are words or non-words and so on. So again, reinforcing this idea that what we can get with resting state functional connectivity are valid measures of the functional organization of the brain. And we can do this across the whole brain. So one of the first things we did, one of the first ways in which we applied this method was to, I suppose, demonstrate its validity. We wanted to see, could we pick up on the same kinds of functional distinctions within and between brain areas that had been demonstrated by task-based fMRI studies. And so we looked at regions like the anterior cingulate, the striatum, amygdala, posterior medial cortex. That was something led by Daniel Margulies. And what we found is, again, these functional distinctions within and between areas, as picked up in relatively small numbers of participants, simply lying in the scanner resting, mapped on really well to what had been demonstrated across hundreds of task-based studies with thousands of participants. And this was really gratifying, and it really validated this approach, so this really simple approach for mapping out large-scale brain circuitry with relatively little data. You're able to map out many, many different circuits, where with a task-based approach, you can really only map out one function at a time. So this is an incredibly powerful approach that can be applied to many different populations. And I think anyone who's familiar with this, with the resting state approach, will know many of the different directions in which it's been taken since. So for example, by Daniel Margulies, who has mapped out functional gradients, gradients of interactions between different functional networks in the brain and a cortical hierarchy, or Thomas Yao, who has mapped out these canonical brain networks, or even Pierre, who has used these data-driven clustering approaches to identify the fundamental building blocks of brain functional organization. But one of the first directions that we wanted to take it was to look at development. So in this really early study, which has an embarrassingly small sample of about less than 15 children, 15 adolescents and 15 adults, something like that, we were really interested in looking at developmental change. So just cross-sectional, how do these networks develop from childhood to early adulthood? And just to focus on one particular network here, the network based in perigenual medial prefrontal cortex, which is a key node of the default network, which you may have heard of as being central to the integration of current cognition with information and memory. What we saw as we looked from childhood to through adolescence to adulthood was a significant change in the organization of those brain networks over time. So while the networks of children were dominated predominantly by short-range local connectivity, the networks of adults featured much more long-range connectivity. And that's also represented in this histogram of the number of significant voxels at different distances from the seed. So again, the children's networks dominated by short-range connectivity, and the adult networks dominated by these more long-range connections. And this was really nice because it mapped on to what had been shown with task-based imaging studies, where you get changes, you get much more diffuse so non-task relevant activation in younger kids that becomes much more specific as you look at adults, and also what the neuroplastic changes that we know are happening on a structural level. So, notwithstanding some very serious concerns about the role of motion, so we know that participant head motion also has these kinds of effects on functional connectivity, and there was a kind of a big period in the field where everybody was obsessed with motion and was really worried that none of these effects were true and would hold up. That has kind of somewhat been resolved, and there is acknowledgement that there are significant developmental changes over time, and these have been replicated and expanded in many subsequent studies. We know that there are significant changes in brain functional organization that happen over development that we can detect with this functional connectomics approach. So why is this important? So I'm going to step back from the data for a moment to just talk about my kind of approach or philosophy here. So I don't think there's anybody maybe in this room who would disagree that psychiatric disorders are brain disorders. And I think it's also quite maybe simplistic at this point to say that psychiatric disorders clearly have their roots in childhood. And this is, this is true not only for the prototypical disorders of childhood like ADHD and autism, but we also know, for example, the median age of onset of anxiety disorders is 11. There are many prodromal signs of schizophrenia that are detectable in early childhood. And even with depression, we know that adolescents who have even subclinical levels of depression are much more likely to go on and develop a full-blown disorder in their early 20s. So looking at development is really key to understanding the emergence of psychiatric disorders. But nobody seems to put these two together, right? So if we accept these two, that psychiatric disorders are brain disorders and that they emerge during development, then understanding brain development is key to understanding the emergence of psychiatric disorders. So what we have with functional connectomics then is an approach that has the potential to provide us with something akin to the pediatrician's gold standard for detecting problems in development. So something like normative growth curves describing expected developmental trajectories, in this case of brain function, where pediatricians use head circumference, height and weight and so on. Having these normative growth curves for brain function would allow us to detect kids who are having problems in development, who are exhibiting difficult or abnormalities or alterations in their brain development that are suggestive of a particular disorder. And this would allow us to intervene at the earliest possible point and potentially redirect their trajectory back towards health. So Nico Dozenbach and colleagues were the first to really produce something akin to a growth curve for functional connectivity. This was using cross-sectional data and about 240 children who were aged 7 to 30, and they used machine learning to create this functional maturity index, which was predictive of chronological age. There was then this kind of hiatus where everybody was very worried about motion, but in the last couple of years, papers have started to emerge that are demonstrating that these kinds of growth curves can be used to potentially detect psychological disorders. So in this study by Kessler and colleagues from 2016, they used the Philadelphia neurodevelopmental cohort. They had scans from over 500 youths aged 8 to 22, and they derived these normative growth curves for brain functional connectivity and found that deviation from those curves was associated with ADHD symptoms. And the similar approaches have been used more recently by Maria Jabrzybowski in psychosis. So really suggesting that this approach provides a valid way for us to detect problems in development that are indicative of psychiatric disorder. And what's even more exciting than that is the fact that we can do these functional connectivity, we can collect resting state fMRI data and do functional connectivity analyses from in utero. So Mariah Thomason, who's now at NYU, is one of the pioneers in this space, collecting data from 25 weeks. So you can imagine the potential to really have these normative growth curves charting expected development from in utero to add it to all the way to the other end of the life cycle, because these methods are so accessible, and it really gives us a universal approach to study brain development and how that goes awry. Okay, so the goal of all of this, of course, is to develop better biomarkers, right? So we want to find predictors of disorder, and that's the goal of everybody working in biological psychiatry. To have clinically useful biomarkers, we need measures that have high validity, reliability, sensitivity, and specificity, and that's actually quite a large demand that we, in many cases, don't really meet, as I'll kind of talk about in a few minutes. So one of the things that biomarker discovery really requires of these large data sets. We need enough data set, we need enough data to have like sufficient variance in the disease of interest, but also to have other diseases in there so that we can really assess things like sensitivity and specificity. And resting state data is perfect for this challenge, right? Because it doesn't involve any particular paradigm, we can pool it across labs. So data collected here in Montreal can be pooled with data I collected in NYU or in Dublin or wherever. We can analyze it all together and we can really create these enormous data sets. And I think at this point, maybe, you know, people who are newer to the field, these kinds of enormous data sets are very much the norm now. But the kinds of data sets that we have with several hundred or 1,000 participants used to be were the stuff of dreams when I started my postdoc. And so the power that we have now to try and create or identify these biomarkers is really impressive. And so some of these efforts have been yielding really great insights into different disorders. So for example, the ABIDE data sharing effort that was led by my colleague from NYU, who's now at the Child Mind Institute, Adriana Di Martino. The first round of that involved over 1,000 participants. There's been a second round since with another 1,000 participants. And with this kind of sample size, Adriana was able to reconcile disagreements and inconsistencies in the literature about the nature of brain-based abnormalities in autism. So where both hypo, or too much and too little connectivity had been identified, she was able to separate out, that hypo connectivity or reduced connectivity was primarily between cortical areas, whereas hyperconnectivity was limited to subcortical circuits. And this really helped resolve a lot of the questions in the literature. And of course, these data sets have been used by many, many different studies. So here's an example from Suk-Jun Han, led by Boris Bernhardt, who's here in Montreal too, where they use the ABIDE data set and Daniel Margulies, his nice cortical gradient analysis to show that there were atypical transitions between functional networks in autism, suggesting an abnormal cortical hierarchy and an imbalance in the communication between these networks. And these abnormalities actually correlated with some of the behavioral deficits in autism, including social cognition and restricted and repetitive behaviors. So to turn to another disorder and the kinds of insights that functional connectivity analyses have yielded, this is a nice paper from Drysdale et al., again combining data collected across many different sites to detect biological subtypes that they call biotypes of depression. And what was really nice in this study is they found these 4 different biotypes, so different patterns of functional connectivity abnormalities that mapped onto different kinds of symptoms in the disorder, so different combinations of anxiety, anhedonia, fatigue, and so on. And what was really exciting was that these different biotypes also were differentially predictive of treatment response. So this is all really, really promising and really great. There's always a but, right? So everybody was very excited about the Drysdale analyses, but just recently, just this year, a failed replication was published in Neuroimage Clinical. So in a more heterogeneous sample, so obviously it's going to be a bit more challenging to do a replication, this group failed to find significant biotypes. They just completely failed. They replicated the analyses. They did find biotypes. They were not statistically significant. And in this study, looking at again the ABIDE sample, just highlighting some problems of the fact that while you can detect ASD-related abnormalities across sites, and you can detect them within sites. If you try to look at the reliability of those or the similarity of those ASD-related differences detected, if you compare them across or between sites, there is not good similarity in. They don't replicate. So this is a bit of a concern, particularly for these multi-site and probably for these more retrospective studies that have combined data that were collected under completely different protocols with different behavioral measurements by completely different labs in a very unorchestrated manner. And of course there are huge, much more orchestrated and careful projects ongoing at the moment that use functional connectivity or resting state data, like UK Biobank or like the Adolescent Brain and Cognitive Development Project, that hopefully won't face these same challenges because they're going about it in a much more careful and measured way. But one of the, I suppose I should talk now about what is the approach that I've been taking to these challenges and these kinds of data. So I suppose one of the things that's worth saying is that we still haven't got any neuroimaging biomarkers that seem to be reproducible, that seem to have good diagnostic or prognostic validity. Those have not, even though this field and this method is hugely promising, we still don't have anything that's even close to being considered a biomarker. And one of the reasons for that, of course, is heterogeneity within and overlap between diagnostic categories. So we know that, for example, autism is an enormously heterogeneous disorder. So I could have two kids who both have a diagnosis of autism who have completely separable, non-overlapping difficulties. The same is true in depression and virtually every disorder. And then equally, disorders have a huge amount of overlap. So autism and ADHD are often very difficult to tell apart. Kids with autism and kids with ADHD often have comorbidities like anxiety. And it's really hard to tell when you identify brain-based differences in these groups, what's attributable to the autism versus the anxiety versus ADHD and so on. Part of why we need huge samples, but the other problem is that many of the lab-based measures of cognitive function that we use to detect, dysfunction or disability in these disorders have pretty poor construct validity. And some of this might actually just reflect poor ecological validity. So I'm sure you can think back to maybe some psychological experiments that you've taken part in, maybe imaging experiments, where the task is, pretty boring. It's not, you're doing it because the experimenter told you to do it, but you don't really get why. And you can imagine that kind of situation is worse for somebody who has a neurodevelopmental or psychiatric disorder, where, we can kind of make this attribution that, well, the experimenter knows what she's doing, so I'll just do what I'm told. I'm sure it makes sense, but somebody with a disorder might not make the same inference. And so that could really affect, the kinds of behavior that that the tasks reveal, as well as the patterns of brain activation and our ability to detect relationships of connectivity. And it was just kind of lucky that yesterday Simon Eickhoff, I don't know if people here would know him well, but he is a very famous functional neuroimager. He posted on Twitter about, he was posting a link to this nice new task, and he was just saying this is a really nice example of a more ecologically valid measure, in this case of motor intention. And games and movies should be more engaging and closer to what the brain evolved for. So just emphasizing this problem of ecological validity. And this is something that we've been trying to address in the lab. So we really want to refine the approaches that we use to measuring ability and disability in the disorders that we're interested in. And so we've been particularly interested in ASD, autism, and adolescent depression. And we really want to find cognitive and behavioral dimensions that are more sensitive and specific to the patterns of ability and disability in these disorders. And we also want to use approaches or measures that have more translational validity, so that have maybe animal analogues, so that we can really link some of the advances that have been made using preclinical or animal models to what we are doing at the human level. So this is where I start getting into some of the much more preliminary work, and in some cases I'm just going to describe some of the tasks that we've used, and I don't actually have any results yet. So this is one of those cases. So this is a project being carried out by my PhD student, Anna McLaughlin, and she's really interested in autism, and she's particularly interested in the social motivation hypothesis, which suggests that autistic individuals experience just decreased reward from social stimuli, and this is why they're less motivated to engage socially. Loads of studies have compared response in autistic individuals to social versus non-social rewards. So social rewards like smiling faces, non-social rewards like money. And the results have been really inconsistent. So some suggest a specific deficit, or a deficit in reward processing that's specific to social stimuli. Some suggest a more global deficit. And actually a recent meta-analysis came down on the side of a domain general reward processing deficit. But this conclusion is pretty much at odds with the restrictive interests that characterize the disorder. So it's one of the major symptom domains. Autistic people clearly derive pleasure from the interests that they have. And so that it doesn't make sense then to say that there's a domain general reward learning deficit if they clearly have preserved reward learning function in some regions, in some domains. So what Anna has done in her project is to incorporate specific objects of idiosyncratic interest into a reward processing paradigm to try and better get at this idea whether autistic individuals have reward processing abnormalities or not. And so, and this is not unique to our lab. So Rebecca Jones, for example, in Cornell, has also been doing this in her work in autism. So we created this simple reinforcement learning paradigm that had three conditions. It had social rewards, it had monetary rewards, so social rewards were just smiling faces, monetary rewards, and then preferred stimuli. So what we did was we created this massive set of pictures of nice things, and we did poll some autistic adults first to get a sense of what people's individual interests were. And so what we do at the beginning of the task, we show lots of these pictures, and we get them to say get the participants to say whether they like the picture or not. And then we take the kind of most liked categories and use them in the preferred stimuli condition. And we have this really simple task that's a probabilistic reinforcement learning task, but it was designed to be a little bit less abstract for participants. So essentially what participants have to do is learn a reward association that's probabilistic. So in one condition, these are two doors that they're told they have to open to find a reward. In one condition, one of the doors is associated 100% with a reward and the other 0. And then in a different condition, it's 70-30. So they have to, obviously in that condition, it's more difficult to learn the reward association. So we tell the participants, you see two doors, you have to open you have to pick one to open to try and find a reward. Sometimes the doors get stuck. And if it gets stuck, you won't get to open it to receive the reward. So try to avoid the sticky doors. So the idea is that this would make a little bit more sense to people as they take part in the experiment. And then the rewards were, like I said, these different conditions: the preferred stimuli, a social smiling face for the social stimulus, and then monetary reward. So this study is very much still ongoing, so I don't really have anything to present at this time, but hopefully we'll get this finished up by the end of the year. So another thing, like I just mentioned, something that we really want to try and figure out in autism is this anxious or comorbidity with anxiety. So it is thought that about 40% of autistic individuals have an anxious, have an anxiety comorbidity. And so together with Mark Bennett, who is a postdoc in Trinity, is now in Cambridge, we wanted to develop, again, a nice, ecologically valid task that could be collected in a pretty short space of time that might give us insights into anxiety and to be able to say something about how these kinds of fear associations are learned in autism. So children with higher trait anxiety show overgeneralization of fear learning, but one of the characteristics of autism is that they're very specific in their learning. So we wanted to see if maybe there's a distinction amongst individuals with autism who are anxious versus those who are not in terms of this generalization of fear. So what Mark did was he developed this really nice version of a kind of novel paradigm. It's called one trial overshadowing, which is literally one trial learning. So in one shot, participants are exposed to like a fear learning stimulus. So they're exposed to a cue that's paired with a loud scream. And then they're exposed to kind of different versions of this. So a solitary cue, a compound cue, and then a safety cue, which doesn't have any negative association at all. And this allows us to distinguish between, or to look at fear learning and generalization in a really short paradigm that's just about 5 minutes. And Mark designed it to be really kind of nice and gamified, so it looks like a little Indiana Jones guy, and you have to make decisions about whether to open this chest to find gold coins, but avoiding the chest that's paired with the danger stimuli, the one that you had heard paired with a scream. So I won't go through the task in much more detail, but just to say it's actually working really nicely. We're getting this really nice distinction just with this one shot learning. People are learning the fear association, and some people are showing generalization to other stimuli. And so what we're hoping is when we look at those who have high anxiety symptoms, then we'll be able to really we'll see differences in terms of the performance on this task. Okay, so I realize that I'm talking through a lot, so I might skip over some of the next few slides because I really want to get to the translational stuff. But one of the other disorders that I'm really interested in is adolescent depression. This is a project that was really carried by this really incredible undergraduate student who's now a PhD student in the University of Edinburgh, Niamh McSweeney. And I'm hoping this data will come out in a paper in the next couple of months. But we know right now that depression is the leading cause of illness and disability worldwide. So that's more than cardiovascular disease, more than cancer, more than any other condition you can think of. Depression is the #1 cause of disability and illness. And it is the case that from age 13, females are diagnosed with depression at twice the rate of males. And like I said earlier, many of the adolescents who experience even subclinical symptoms will go on to develop full-blown disorder. So what we're really interested in is what are the kind of psychological and neurocognitive features that differentiate individuals who struggle with depressive symptoms during adolescence from those who do not, who are more resilient. And we were really interested in coping styles. And this is partly driven by the fact that coping in humans is entirely assessed on the basis of self-report. So people are just asked about how they cope with different situations, and we don't really have any objective measures. But Daniela Schiller of Mount Sinai and colleagues have developed this really nice human translation of an animal paradigm called the Sidman avoidance paradigm. That allowed for the assessment of objective assessment of coping. So we looked at this paradigm in a sample of adolescents with depressive symptoms. So they didn't necessarily have a diagnosis of depression, but they had depressive symptoms. And what participants have to do is they have this very simple game board on the computer, and they have to learn to move their cursor from one side of the board to the other, because this is maps on exactly to what the rat has to learn. And in the case of the rodent studies, the rat receives a shock when they fail to move from one side of the, in their case, the cage to the other. In our case, the participants heard a really loud noise and saw this shock or a lightning bolt on the screen. So they had to learn this through trial and error, and they received 1 point for every time they avoided kind of getting a loud noise or a shock. We didn't shock the adolescents. The shock is what the rodents get. So good performers learn to do this, and poor performers don't. Essentially, and so these are people who have active coping. They figure out the task, figure out what they need to do to avoid the negative stimuli, and they just keep doing it. Poor copers don't do that. They kind of try it a bit, and then they essentially don't respond as much anymore. And this maps on really well to what the rats do, because they just freeze and don't learn to avoid the response. So what Neve found is that the number of active coping responses that a participant exhibited was correlated with depressive symptoms, and this correlation was stronger when we just looked at those with clinically significant symptoms, so with a BDI Beck Depression Inventory score of more than 16. When it came to looking at the brain connectivity, this is still very much a work in progress. So Collins, in the original paper where they demonstrated this task, they found a relationship between functional connectivity between amygdala and medial prefrontal cortex and task performance. We didn't see that relationship, but we did see a relationship between the same areas and depression severity. And that was significant for both left and right amygdala. So we're still, this is a very limited preliminary analysis, and we're still, we need to work with a full sample, and we need to take a much more broader approach to brain connectivity to look at what the kind of correlates of the coping task performance are in this sample. Okay, I'm going to skip a little bit through this next bit, because it's where I was just going to talk about some of the other, with all of the data that I've talked about already, like the autism study, the depression study, we have resting state data in all of those participants, and we're going to look at relationships between the behavioral measures that we've collected, so things like the reinforcement learning or the anxiety, and relate that to functional connectivity. We also, as well as collecting just rest, we have been collecting these passive viewing or movie conditions. So some of you might be familiar with Tamara van der Waals Inscapes paradigm, which is just like, she hates when people say this, but it's kind of like a nice screensaver. So it's something just to look at in the scanner. It's not very evocative, it's not very stimulating, but it keeps you still and keeps you awake. So we've been collecting those data. We've also been collecting this, again, a movie that's like the original Hyder and Simmel paradigm that maybe some of you will be familiar with, which features just moving shapes. And people typically attribute mental states and intentions to those moving shapes, but people with a diagnosis of autism often don't. So they'll say, well, we had one participant say, well, I know I'm supposed to say they're dancing, but those shapes are just moving around the screen. So there's a difference in the kinds of attributions that are made. And we've been looking again at relationships between those kinds of attributions and functional connectivity. But I'm going to skip over all of that because I just want to get to where I would like to go with all of this. So one of the biggest challenges is that we lack a good understanding of what the hell functional connectivity is, what it's doing, what it's for, where it comes from. We don't have a good understanding of its neurophysiological bases. And we also don't understand the mechanisms through which functional connectivity is disrupted during development by any number of the influences. So we can think about stress, for example, or adverse experiences during childhood that may precipitate depression. How do these change the brain? To answer those questions, we have to kind of, I don't think we can do this in humans alone, right? So there are some great efforts ongoing, like the ABCD, where they're looking at following children from age 9 to 19 over 10 years, collecting loads and loads of data. But those are observational studies. To really gain mechanistic insights, we need experimental manipulations. And to truly chart longitudinal, or to chart these normative developmental curves, longitudinal data are needed. And so ABCD will yield those kind of data, but in 10 years. If we think about working with non-human models, then we can collect the kinds of data that we need to derive developmental curves in pretty short spaces of time, particularly if we work with something like a rodent. So for me, the next frontier is really working with translational functional connectomics. So working with preclinical models, particularly rodents, in order to achieve some of the things that we want to achieve. And the reason that this is an entirely reasonable approach is because there's remarkable cross-species correspondence in the functional networks that are detected. So here, for example, is the default network, which for a long time people considered to be a really, human network. It seems to subserve very human-like functions, thinking about the past and future. Like I said, integrating, what current demands are with the contents of memory. Yet it has been very compellingly demonstrated in both non-human primates and in rodents. So we can detect analogs, certainly, of any of the functional networks that we detect in humans in non-human models. So, by doing that, once we know that we can use the same kind of approach, it makes preclinical models really powerful, right? Because we can do all kinds of experimental manipulations that you couldn't possibly do in humans. We can do pharmacological, molecular, genetic, all kinds of manipulations that can give us much more causal insights. And then as well, the time scales of development are obviously much more tractable. The environment is under our control. One pitfall, of course, is that they often have to be anesthetized to go in the scanner, but there are other protocols that can be used that feature more sedation rather than anesthesia. And some people are working with animals who are trained to tolerate the magnet while awake. So it has huge potential. So I'll talk really quickly about this study that hopefully is going to go on bioRxiv in the next couple of weeks. That is the result of kind of a long collaboration with Mars Sanchez, who's at the Emory, the Yerkes National Primate Center in Emory, and Melanie Pinzas, who is a PhD student who has recently graduated. And what we're interested here, again, is this idea that adolescence is this time of huge neuroplasticity, but also considerable vulnerability particularly for females, to psychiatric disorder. And many people, hypothesize that this vulnerability in females may reflect some sort of interaction between adverse experiences, so exposure to stress, for example, and then normative adolescent sex-dependent adolescent changes in, for example, the levels of sex hormones. So this study, in this study, we looked at whether chronic stress and puberty have separate or interacting effects on brain functional connectivity and behavior during adolescence. So the participants were these socially housed adolescent female macaques. And the thing about them is that, so in the Yerkes National Primate Center, they have these huge enclosures, kind of like a zoo. They have these big enclosures where these animals live in fairly normal conditions, within the restrictions, obviously. But in this kind of environment, the animals, the females, organize into a social dominance hierarchy. And this provides a really nice model of chronic stress, because if you are a dominant animal, that's great, you're at the top of the pile. If you're a subordinate, you're subject to fairly random and constant harassment. And so it provides a really nice model of human psychosocial stress, so the unpredictability of human psychosocial environments and so on. The other nice thing about it is if your mum is a subordinate, then you're a subordinate too. So from birth, you're really exposed to this stressful environment. So to kind of separate the effects of stress from the effects of puberty, in half of the sample, Mara and colleagues administered a puberty blocker. So what we wanted to do was see if experimentally delaying puberty onset would be protective, because some theorists like Charles Sisk and colleagues have proposed that the brain becomes less sensitive to the effects of sex hormones with age. And so we thought that if we delayed the onset of puberty, then subordinate animals in the delayed group would not show the same effects of stress as the ones in the normal puberty onset group. So what we were looking for really was this interaction, right, between the rank, so the monkeys' rank in the social dominance hierarchy, and whether or not they had puberty delayed. And so we had, I'll go through it quickly, about 30 monkeys in each group, which is really well powered for an animal study. But the data were all collected after the experimental delay of puberty had ended. So at this point, they had all gone through puberty. That may explain why we didn't really see the strong effects that we expected, and we didn't obtain any of the interactions. So what we essentially saw were just separate effects of the stress in terms of the social rank and the experimental delay of puberty on both behavior and on brain connectivity. So, for example, we saw that more stressed animals showed weaker functional connectivity between amygdala and dorsolateral prefrontal cortex, whereas the delayed puberty animals showed stronger connectivity between amygdala and orbitofrontal cortex. So we saw these very separate effects, and we also saw the separate effects on behavior. So the chronic stress was associated with negative effects. The delay of puberty seemed to have positive effects on some behaviors and some negative effects on others. These were very separate from one another. And so there wasn't really this evidence of a neuroprotective effect of delayed puberty that we hypothesized. And instead, it seems like this experimental delay of puberty just had yielded a kind of developmentally immature phenotype. It may be that we were quite underpowered, and it probably means that we just need longitudinal studies to really pull apart these effects. So it was a really interesting study. It has yielded really good data, but wasn't as conclusive as we hoped. I'll finish by just saying that I really want to essentially do the same thing in rodents, and that's where my work is going next, along with collaborators from Trinity who are all experts in rodent models, particularly of depression. And I'm hoping to map longitudinal development in rodents using functional connectivity to yield those kinds of developmental curves, and then impose a stress manipulation and see how that stress alters functional connectivity during development and how that differs between males and females. So I will skip forward to the end because I have talked for a long time. I just wanted to highlight the fact that there are open data from animals available. So Mike Milham, my former mentor from NYU, is at Child Mind Institute, and who's a pioneer in all of this stuff. I'm not sure that all of the open data that there are would exist without his original 1000 functional connectomes effort. So they have released a load of data from macaques, male and female. I can't remember, I think it's something like 60 or maybe more than 100. So that's potentially really powerful. And then there's also a mouse, so there's data, a mouse multi-site study. So again, involving, I think, over 100 mice collected in lots of different centres that's available on Open Neuro. And I know there'll be more data later this year from Valerio Zerbi in ETH Zurich. He's going to release 100 mice as well. Okay, so I hope I've given you a sense of the power of this functional connectomics approach, the kinds of insights we can gain, but also some of the kind of limitations and things that we need to do to really move forward with the goal of detecting clinically useful, sensitive and specific biomarkers. That we need these open data sets, we need to keep doing that, we need to keep collaborating, we need to keep working on these huge data sets, but we also need to hone the measurements that we use. We need to figure out better behavioral dimensions that can help us to identify the dimensions that like map onto patterns of brain function and dysfunction. And to really get mechanistic insights, we need to think about translational approaches. So thank you for listening to me for such a long time. And Pierre has asked me to give a more introductory talk to functional connectivity on Friday. So if all of that was not very clear, hopefully I'll make it a bit clearer on Friday. Thank you. I'm sorry for talking for so long.
Thank you. For what? Perfect. Are there some questions right away? Maybe I'll start to warm you up. You mentioned the macaque study with social stress, which is like really cool. I don't know, is that already published?
No, we're trying to get it hopefully on bioRxiv in the next kind of 2 weeks. It's been
amazing.
4 or 5 years on the go.
I'm sure. It sounds very complicated to deploy. How many monkeys do you have in there?
I think the full sample was about 30 something in each group, but with quality control and various other issues, I think it's down to 20 something in each, in the experimental delay and the normal puberty, and then the social rank is continuous. So we were looking for an interaction between a continuous variable and a categorical variable. So I think we were quite underpowered. But
do you have a feel of the inter-individual variation in monkeys? Because in humans it's huge.
I think it's pretty big, yeah. And then particularly when you have something like this where there's a clear inter-individual difference, right, related to the environment that they've experienced growing up. That's what's really nice about that study, is that they've group living monkeys, which it's unusual. That often many of the primates that are used, non-human primates that are used in these studies, live in quite experimental, the cage kind of conditions, whereas these are quite. I had a picture, didn't I, in one of the.
So where did you find them? They were just like hanging out in the forest.
Oh no! This is the Yerkes primate. I mean,
you had a gnat on a tree, and this happened to respond.
So it's not. This was all done by Mara and her group, and I've never been there, so I don't know. So these are chimpanzees, right? So this isn't accurate, but this is what it does look like. They live in these this compound, and they have it's an enriched environment. It's quite different to the normal environment that experimental primates have. So I think it's a really nice, and she, like Mar Sanchez, does really great stuff with these kind of more ethologically valid. So looking at individual differences in parenting, essentially, in these macaques, because some of the females just are not good at taking care of their babies, and they're quite mean to them. And so this is it's kind of a nice ethologically valid model of human child neglect and abuse. So it offers a lot of insights that we couldn't get in any other way. Yeah.
I have a question. Do you think if you delayed puberty longer, because it looked like there was an overlap, you think if it just delayed it longer, your effects would be
Yeah, I think it's you know, I didn't have anything to do with the planning of the design or anything.
Sure it's very complex,
Yeah, like
it's
this is what they do. So there's Mark Wilson is actually the other PI on this, and he's a neuroendocrinologist, and so this is, you know, his study and stuff. And it was just, I wrote like a small grant to reanalyze data they had collected as part of the study. I think that one of the reasons we didn't see the effects, we didn't see as great an effect as we expected, is that when they were imaged, they had all gone through puberty because the drug had been stopped. And so it probably washed out a lot of the effects. I mean, that in itself is interesting, you know, but it's just, we're just really at the beginning of this kind of stuff. And I think there's a lot of potential to expand that study to rodents, where you would have, you know, it might be a little bit easier to control and to look at things like, how long and maybe get more fine-grained. You know, this was one time point, so we're not even looking at changes or anything. It's quite limited, but I think really interesting.
Do they grow more? Because you, when you are in your young age, you grow, and then when you have the puberty, you stop growing. Do they grow more?
That's I don't know the answer. Sorry.
Okay.
Yeah, and it's interesting. It's not even in the paper, so I might flag it to Mara and ask her. I'd imagine not. Because these were females as well. So with males, maybe they would grow a lot more, but it's a really good question. I'll try to find out.
Okay, I have another question. Sonia Lupien made studies on, you know, young that learn at school. They were able to learn, but some of the kids that were beaten by their parents, they don't learn much.
Mm-hmm.
But then they realized they were having other abilities. You know, they make they blur images of mad people, and they will recognize mad people even if they were blurred, you know. But the other kids were not able to do that.
Yeah.
So they learn different abilities to survive.
Yes.
So maybe
they
have a different resting state.
I'm sure. Yeah. I mean that their threat, you know, their threat detection is better than kids who haven't had that experience, and that makes sense. And that's why I think it's, you know, it's really important that that we think about all these different behavioral domains, that, you know, and not just think always about disability as well. You know, even though that ability comes from, I suppose, a bad place, it's still something that they're strengthened on. So yeah, I think I'm sure they would absolutely have differences potentially in you know, limbic or amygdala functional connectivity that might be related to that, and I'm sure other behavioral differences too. Yeah.
Do you see that where your monkey, that the mother was not good with him?
So Mar, I can't remember now. I worked a little bit on some data that Mar had from these abusive mothers, but I can't remember if that ever got published. So I must have a look. But Mar, if you look at Mar Sanchez from Emory, so she has lots of work in this area.
All right, so maybe we're going to transition to the free hacking anyway. Claire is here for another couple of days, so don't hesitate. I think we should thank her again.